Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the molecular basis of inheritance. This is a part 1. The chapter 5, the molecular basis of inheritance. Molecular basis of inheritance. Let us start. See, the previous chapter you have learned the inheritance pattern and the genetic basis such as any patterns. Means the principles of inheritance we discuss about as in how to the genes will be they carry from as in one generation to other generation. We learn in that in previous class. Am I right? Here, at the time of the Mendel, the nature of those in factors regulating the pattern, the inheritance was not clear. So what the Mendel they explained that all characters they are not they have clear because those time what happened they're completely the compound microscope is not available. Am I right? Like, and over the next 100 years here the nature of the primitive the genetic material the investigated the accumulating that in the realization of the deoxyribonucleic acid that is in a DNA, it is a part of what genetic material. So after 100 years we noticed as in when primitive the genetic information they get carrying from as in one generation to other generation, we identified that base is what that is in a genetic material DNA, that is the deoxyribonucleic acid. Let us at the last for as in that majority of the organisms they having what the DNA is present. And in class the 12th you have learned about that is the nucleic acids in the polysomes and nucleotides. So in this 11th class means that is on a biomolecules chapter we discuss about as in what is the nucleotides and nucleic acids, so we learn already. Yes, now here the deoxyribonucleic acid the DNA and the ribonucleic acid RNA. So these two are completely the nucleic acid to found on the completely the living system. So already when you take as any one particular living system, the living system they contain as what the genetic material is present. The genetic material that is either in a DNA or RNA is present. Am I right? Let us here the DNA act as a genetic material in most of the any organisms, and RNA through as an also act as a genetic material in some viruses like. So here the living system they contain as in that mostly in what DNA is present, but acellular organisms as present like as in a viruses is there na. So viruses is contained as what that is in a virus contained as in a RNA means ribonucleic acid the genetic material is present. Next, mostly the functions as in the mRNA. So RNA get act as what it act as another messenger molecule means to get pass the message from as in a one DNA to other DNA. So that is in a messenger molecule. And RNA additionally their role what is that ma that is in a they participate in different functions like as an adapter molecule, okay, and messenger molecule like. See here the functions as in the adapter molecule, the structural gene, and the some cases they are the catalytic molecule also. So here RNA it act as a catalytic molecule also. Now here in class 11th you have already learned about as in the structures of the nucleotides in the way of these monomer units that are linked with form as a nucleic acid polymers. So the last previous class means in that biomolecule chapter we learn about as any the completely the nucleotides. We learn about as any nucleotides. See here I draw one the diagram of any the nucleotide. That is what it is in a pentose sugars. It is in a the pentose sugar, and it is in a phosphate group. It is in a phosphate group, and it is what nitrogen base. Already you know that nitrogen bases they have in the different types. Am right? They are like as in adenine, guanine, thymine, uracil, cytosine. Yes, these are the completely different types of the nitrogen bases. Let us here this chapter we are going to discuss that structure of the DNA. So what is the exactly structure of the DNA? They complete the structure of the DNA, and here the process of the making of RNA from DNA. So the total process called what there is a transcription process, and the genetic code that is determines the sequence of the amino acids means how to the completely you take as any one particular the amino acid will be takes place in this amino acids of where they're binding. So the binding site based on the proteins we discuss, and the process is called as what? Protein synthesis process, that is in a translation, and then elementary basis of the regulation. These are completely we discuss in that in chapter the molecular basis of inheritance. Okay, and then the determine the DNA complete the nucleotide sequence the human genome during as an during the last decade, the set of the new the genomic that sections they are essential for as an that human genome sequencing and consider will as in that discussed. So how do the genes they are carrying from as a generation to generation, and what is their functions, how do this total process will be takes place, we discuss here. Okay, let us begin our the discussion. The first, the understanding the structure of the most the interesting molecule, that living system. That is what, Nana? The most interesting the molecule is what? That is in a DNA. That is what? That is in a DNA. That is, in the sequence of the sections will be the understanding why it is the most abundant genetic material and what is the relationship with as an RNA. So, what is the structure of the DNA and how do they get to get form the DNA strand, and what is that the connection between as an DNA RNA? Let us we discuss. Okay, yeah. Next, here first the DNA. DNA means what, Anna? DNA are nothing but as a deoxyribonucleic acid. Am I right? DNA are nothing but as a deoxyribonucleic acid. Let us. The DNA is completely, when you observe here, DNA is in a long polymer of in a deoxynucleotides. It is a long polymer of deoxynucleotide. The length of the DNA, the usually defined as the number of nucleotides, are nothing but as an a pair of the nucleotides refers to as an the base pairs. So, in this total length of the DNA, they are depending upon the how many base pairs as present here. How many the base pairs as present, they are easily to get calculate the DNA, the total DNA. To get calculated as in the total DNA. Next, here this is also the characteristic the characters of any organism. So how many bases as present, they clearly they give as in the particular information, right? For example, they given here. See, when you observe as in some bacteriophages is there, the bacteriophages the length how do you get calculate? You get calculate as in a here mu naught into 174. It has around has how many nucleotides will be coming here? 5386 nucleotides are comes. And then here the bacteriophage is there. It is a lambda phase. It contains has how many? 48,500 base pairs as present here. And you see as in a next in that Escherichia coli how to get calculated then total the DNA length it is in a 4.6 into 10 to the power of 6 base pairs. It is what? 4.6 into 10 to the power of in a 6 base pairs. Let us see here. When you observe to as in that completely their haploid content of the human DNA, that is in a the 3.3 into 10 to the power of 9 base pairs. Now I am asking you, if in case I given here only for the DNA haploid, now you get calculate the diploid means what, Anna? It is in a 6.6 into 10 to the power of 9 base pairs. Am I right? Because this is in a haploid and this is in a diploid DNA, that we can easily to get calculated the length of the DNA. Okay, let us we discuss about as a structure of any polymer. Let's we start with as in what? Structure of the polymer. Yeah. Yes. Let's run here the structure of polynucleotide chain. Structure of polynucleotide chain. Let us here the replicate that in a chemical structure of in a polynucleotide chain, that is in a DNA either in a RNA. A nucleotide has in the 3 components as present. They are what? The 3 components are completely nitrogen based, pentose sugar. The pentose sugar they have in a ribose sugars, the ribose sugars as present in that RNA, and deoxyribose sugars as present on the DNA. Let us only we discuss this one, right? Here I draw here one completely one nucleotide. This is one nucleotide. It is in a pentose sugar is present, and it is in a it is a phosphate. It is in a phosphate group, and it is what? Nitrogen base. It is what, Anna? Nitrogen base is present, and here most of this there are two types of nitrogen bases as present. One is in a purines. See the nitrogen bases. Here the nitrogen base is present now. The nitrogen base is again to divided into as in a two types. One is in a purines. One is for as in what? In a purines. The second one is what now? That is in a pyrimidines. That is in a pyrimidines. Here what are the purines? Purines are nothing but adenine and guanine. Here purines are nothing but as what now? That is in a adenine as well as in a guanine. Adenine and guanine. And pyrimids are what? That is in a cytosine, uracil and then thymine. Uracil, cytosine and then thymine. These are the completely in a pyrimids. Let us. Cytosine is the most common for both for DNA and RNA. See here, most of the cytosine is present is there. The cytosine, it is completely both as present as in a the DNA as well as in a RNA. Okay, and here thymine is present in only for DNA. Thymine is present only for the DNA and uracil is present only for RNA instead of what? Thymine. Instead of what ma? That is in a thymine. Next, when you observe here, the nitrogen base are linked to as an OH in a first carbon, the pentose sugar through as an N-glycosidic, here N-glycosidic linkage from as a nucleoside, such as called as an adenosine or deoxyadenosine, guanosine or deoxyguanosine, cysteine or deoxycysteine, uridine or in a deoxyuridine is present. See here he explained about as what? See here, mostly it is in a pentose sugar. Am I right? In this pentose sugar, it is in a nitrogen base. So between this complete linkage is called as he say about as what? That the enter the linkage is N-glycosidic linkage. There is what? N-glycosidic linkage. Already you know the nitrogen bases. The nitrogen bases are nothing but as what? Their adenine, guanine, they are completely cytosine, uracil and thymine is there. So if in case which nitrogen base is present, we consider like only. Okay, for example, they have adenine is present, that is in a adenosine or deoxyadenosine. Adenosine with the RNA, deoxyadenosine means that is in a DNA. Am I right? And here guanosine, deoxyguanosine, guanosine, here they complete the part of what is the RNA, it is in a DNA. Like cysteine and deoxycysteine means here what they are explaining about here which organism means RNA is present as well as in a DNA is present. So each and every the nucleotide they get followed by as in a the different they are chemical constituents. Okay, let us. So this linkage is complete. It is what? It is in a N-glycosidic linkage. It is what? N-glycosidic linkage is complete. Let's see what Nana after. When see here, when the phosphate group is linked to as an OH group, here when phosphate group is linked to as an OH group and in a 5 end, see here it is in a fifth carbon part. In this fifth carbon part they having as what? It contain as in a phosphate group is present. They are complete the nucleosides there through as in a the phosphoester linkages. So here what is present here is a phosphoester linkage is present. This bond is called as what? It is in a their phosphoester linkages. It is a phosphoester linkages as present. Okay, let us. After the corresponding the nucleotide or deoxynucleotide and depending upon the types of the sugar present. Either it is in that DNA that is in a deoxy. If in case that is RNA that is in a ribo. Am I right? That is. So that kind of the complete nucleotides are formed. Let us. The two nucleotides are linked together. It is as in a complete 3' to 5'. So for example, this then one complete the chain will be get formed. Here completely one chain is formed. In this completely chain, one strand they can start with as in a 5' to here 3' to 5'. That phosphate diester linkage to get formed as in a dinucleotide. So when this enter the chain will be to get formed, that is called as what? That is in a dinucleotide chain. And more nucleotides can be joined such as in a manner to form as in a polynucleotides. Here for example here I took as only one. If in case this kind you take as a many, means there are more than 6 or 10, you take what? That is enter the chain is called as what? That is a polynucleotide chain. Okay, let us here a polymer thus formed as a one end free. So they given as in one polynucleotide chain. Am I right? It is in a 3' to as in a 5'. Here the number of nucleotide bases as present. It is a one nucleotide, it is also one nucleotide, it is also one nucleotide, it is also one nucleotide. So the total nucleotides to get forms as in one chain like structure. So this is called as what? It is in a polynucleotide chain. It is called as in a polynucleotide chain. Here before we discuss what that is a nitrogen basis. Am I right? See here cytosine, guanine, thymine, adenine, like these are completely called as what? These are completely called as any completely nitrogen bases. Next, after in this the phosphate modality in the 5' and the sugars which is refers to as a 5 and the polynucleotide chain. So here 3' to 5'. The 5' it is refers to as what? That is in a completely 5 end of in a polynucleotide chain. Similarly, other end as in the polymer of the sugars has in a the completely OH group is present. It is in a third carbon position. That is what 3'. Okay, that group which has in a the 3 end of the in a polynucleotide chain. So one is in a 3' and the one is in a 5'. Let us the backbone of the polynucleotide chain forms as in a the sugars and phosphates. So yeah, total when you take as in this total DNA, the backbone of the DNA is called as what? Then that pentose sugars as well as in a phosphate groups. And what is the major function of the nitrogen bases? They given here the nitrogen bases to link to as in a sugar motility project to as in a the backbone. So it is to get forms as what? It is to get make as in a what? Nitrogen bases. They give as any stability. They give as what? Stability to as any the DNA. Remaining to discuss in next class. Thank you for watching. Like, share and subscribe.